Assalamualaikum and greeting everyone. My name is Abdul Alim bin Bahrin. I'm a student of Universiti Brunei Darussalam in Institute of Health Science. So for today, I will be covering the topic cancer protective effects of Prunus Domestica and Prunus Salicina, and this is a systematic review. So for this presentation, I will be covering the introduction, the methodology, and focusing more on the results and discussion, and then I will close with a conclusion. So a short introduction. So Prunus Domestica and Prunus Salicina are two main commercially cultivated plums, and plum actually has a high amount of phenolic compound, with different cultivars or variety will have different phenolic content. The anti-cancer effects are actually associated with its antioxidant capacity, where it either directly affects cancer cell or it synergistically help anti-cancer drug treatment. And actually, this is the first review that specifically examined cancer-related effect of plums. And moving on to the botanical introduction, plums are stone fruits or drip fruits that is similar to peach, apricot, and cherry, which has single seed, which is called a pit, that is covered with flesh. Just a brief explanation on prune. So all prunes are plums, but not all plums are prune. And to differentiate between prune and plum is that when plums are dry without removing the seed, it's called prune. But when the seed is removed and then dry, it will be called as dried plum. And moving on to Prunus Domestica, it actually grows optimally in cold winters, short spring, and long warm summer, while Prunus Salicina grows optimally in warm summer with mild spring seasons. And moving on to the methodology part of the paper, where in the study selection, the criteria for the inclusion are as follows. English and Malay language reported are because that's the two main languages that I speak. And then antioxidant properties of both the Prunus Domestica and Prunus Salicina are included, treatment of all types of cancer, and also there is no limitation imposed on their publication. Well, for the exclusion criteria are, for example, studies that use other subspecies of plums, review letters, case study, and so on are not included as well. Study that use plum pomace are not used, and also other benefit of plum unrelated with cancer are not included as well. And here you guys can see that I actually use 5 main search engines, which are PubMed, Scopus, Cochrane, medXRiff, and Google Scholars. And from here you guys can see a capital N and small letter n. And from the legend is capital N actually indicate the total number of paper analyzed, while the small letter n is the paper included for the studies, for the review. And to the right, you guys can see the Prisma flowchart, which actually like the step-by-step on inclusive criteria of the review. And then moving on to the main part of this today's presentation is results and discussion. So first I will iterate about the antioxidant ability first. So plum extract has shown excellent antioxidant ability through DPPH, FRAP, and ABTS radical scavenging assay. And from one of the studies, which is by Dingra et al. 2013, where they used Prunus Domestica peel and flesh, which are extracted with ethyl acetate. And the IC50 value on DPPH activity were as so. And from this, we can see that the extract actually shows strong antioxidant activities, and it actually also significantly inhibits cell proliferation. There's actually other studies that actually shows with the FRAP and ABTS radical scavenging assay, but for this presentation, I will just be including Digger et al. So for further reading, you guys can actually visit my paper. Then next, comparative studies are also done between different stone fruits, i.e. which is peach, pear, and nectarines. And plum actually shows that it has the highest antioxidant activity out of all the others. And from one of the studies by Kim et al. 2012, plum extract of 3 varieties, which are from Formosa, Oshiwase, and Soldum, which is all Prunus Salicina variety, and a DPPH scavenging activity were done, and the IC50 value was as so. And from this, we can see that from the value, they actually differentiate from a previous paper, where they can see that total polyphenol content of plum extract were actually way higher compared to peach, pear, or apple. And also that plum Soldum extract actually has the highest antioxidant activities on all of the assays that they do. So antioxidant activities can be seen from diverse part of the plum fruit. So it's not just the flesh, but it also includes the peel and the pit. So all three parts of the fruits can be seen antioxidant effect on it. And plum pit actually contain the highest antioxidant effect compared to its flesh and peel, where phytochemical compound called amygdalin actually play a very huge significant roles in its antioxidant effect. Where from one of the paper by Kaluki et al. 2012, where they used Mirabelle plum extract, which is a Prunus Domestica, where all three are used, peel, flesh, and pits. And then a DPPH scavenging activities were done, and the IC50 value were also, and it can be seen that pits had the lowest IC50 value, which shows that it has the highest efficacy, which then followed by flash and then followed by peel. And also there is a significant amount of amygdalin was obtained from the pith extract. This actually one of the reason why it results in the highest phenolic antioxidant. And next, the extract also shows that it increased in the absorption and subsequently the antioxidant activities from the oral cavity to the stomach, but actually decreased when it reached the small intestine. So one of the paper by Yu et al. 2021, where they used Prunus Salicina, and it can be seen that from a DPPH antioxidant activity assay, it can be seen like in 4 different situations where undigested plum, indigested plum, but digested by salivary fluid, then digested by gastric digestion, and digested by intestinal digestion. So from this 4 situation, it can be seen that highest bioaccessible phenolics in plum extract first of all were quercetin, pentosidine, quercetin, and cyanidin 3-O-glucoside. And this content actually decreased after reaching the small intestine, which brings back to the statement that it actually decreased when it reached small intestine. And so next, so antioxidant actually play a huge role in anti-cancer effect. So hence why I actually include antioxidant in this review. And so first of all, it actually inhibit P13K or AKT pathway, which will then actually downregulate MC11 activity. And this actually result in inhibition of anti-apoptic factors and also potentiating the pro-survival effects. So it actually helps directly in combating cancer. But on the other hand, it also specifically binds to certain cancer cells, which will inhibit specific kinase that is responsible for regulation of signaling pathway resistance to chemotherapeutic. So it also works as an indirect to helps in the chemo treatment, which I actually mentioned in the introduction. Next, I will actually move on to the cancer part of it, of the studies. So plum extract decrease the cell viability and proliferation of hepatocellular cancer cell line. So from one of the study by Yu et al. 2009, extract of immature Prunus Salicina were used, and growth inhibition on HepG2 cancer cell were observed, where in concentration 1 milligram per ml, 77% were inhibited, while at 2 milligram per ml, 87% were inhibited. So from this, you can see that plum extract actually show a significant anti-cancer activity. And also adding on, it also results in increase in cleaved caspase 8 protein expression, while at the same time decreasing the caspase 10 protein expression. And just as a side note, caspase 8 actually works, which is actually the protein responsible for inducing apoptosis in cancer cells, while caspase 10 is it works oppositely, where it is a negative regulator of cell death. And yeah, moving on. So in breast cancer cell, plum extract has a significant anti-proliferative effect against estrogen-independent breast cancer cell, while also not affecting normal breast cell, which is what we wanted. But it actually is less significant for estrogen-dependent breast cancer cell, where one of the study observed that higher anti-proliferative effects on colon cancer than estrogen-dependent breast cancer cell. So yes, there is anti-proliferative effect, but it is just less significant as compared to other cancer cell type. And another study actually shows that there's a biphasic result for estrogen-dependent breast cancer, where in low concentration of plum extract, it actually increased cell viability, while at high concentration, the cell viability decreased. And this can be interpreted by that phytochemical can be nutritive in low concentration, but cytotoxic in higher quantity. So, and adding on, so one study also suggests that resistant training and dried plum diet can actually decrease C-reactive protein, CRP, and this actually can lower risk of breast cancer. So, so it can be seen that plum can also be used as a preventive measure as well. So for colon cancer, plum extract has been shown to significantly reduce cell viability and proliferation rate in colon cancer, where dried plum actually shown to decrease colon cancer risk inflammatory marker, which example are which are the sickle oxygen radical absorbent capacity value. Second will be fecal bile acid concentration. Third will be sickle microbial enzyme activities. So adding on, plum extract can actually works as a chemopreventive potential for colon cancer as well, where it actually affects the AKT-mTOR pathway and the microRNA, which are the MIR 143, which this actually play a role in cancer growth. And plum extract actually affect this this pathway and RNA. Adding on, plum extract also decrease pro-inflammatory markers such as NF-kappa B, VCAM1, COX2, and INOS expression as well. Moving on to cervix cancer. So cervix cancer has been shown to be inhibited by plum extract as well, where one of the study by Miljic et al. 2016, plum, Prunus Domestica, wine were used, 3 different varieties of it were used, and it can be seen that from the value of the IC50, it's so, and it can be seen that plums wine actually show a significant cytotoxic effects on all of the 3 cancer cell line used, which are the HEP2C, RD cells, and LTOB cells. So plum extract actually does show an anti-cancer effect on other cancers, such as gastric cancer, leukemia, lung cancer, neuroblastoma, and rhabdomyosarcoma, but it is not as prominent as the other cancer type that I mentioned, the 4, for example, like hepatocellular, cervix, and so on. So to end my presentation, I would like to close it with a conclusion. So this systematic review, first of all, is actually analyze relevant literature on anti-cancer effect, and which includes the antioxidant effect. And also that most of the study review on this paper are actually in vitro, while only few were in clinical studies and in vivo. So therefore, it will be actually beneficial to perform more studies on animal or human to actually confirm the result obtained from this in vitro studies can be extrapolated in a wider range of application. Adding on also, studies on lung cancer should be actually further investigated using prune extract as well. And this is actually the reference for the table value that I actually present today. And yeah, thank you for your time. Thank you for your time for listening. And this is actually the QR code for the paper that I'm presenting for today. So feel free to dive in a bit more for the reading. Once again, thank you very much, and